am Manisha. Today I am going to talk about the paper that was published in December 2024. The title is Accurate Sample Deconvolution of Pooled Single RNA-seq Using Sex-Dependent Gene Expression Patterns by and all. So to begin with, some background about single RNA sequencing, that it allows profiling of the cell type specific gene expression. But for doing the single nucleus RNA sequencing, the sequencing costs are really high, and also the limited sample material often because we have limited sample material, we often need to pool the nuclei from multiple samples. So we need to demultiplex or deconvolute post after the sequencing. This is basically to reassign each nucleus to its original sample after pooling. This is to maintain the biological interpretability. There are two existing methods, the already existing methods for deconvolution, known as barcode hashing and genotype-based demultiplexing. But even these methods have some limitations. Like barcode hashing, it requires additional sample preparation, while the genotype-based method requires you need to prior genotype your data. In this particular paper, they propose a solution to this problem. They are utilizing the sex-dependent gene expression patterns to deconvolute without extra processing or without needing any extra experimental or the sample barcode information or extra processing steps. So the key idea for this particular paper is this is for sex-specific gene expression. They try to detect in single nucleus RNA-seq data. In this approach, they pool one male and one female per sample. And in this method, they are using the machine learning models to classify cells sex based on the gene expression. Also, in this, the novelty of this particular method, the authors have proposed is that no extra experimental labels or sample-specific barcoding is required, and therefore reducing the cost and the complexity of the whole process. So, just a brief overview of the methods. The authors use their previously published single nucleus RNA-seq dataset from the rat VTA area, ventral tegmental area, and it consists of over 22,000 cells. So in this workflow, basically, they do the pre-processing of the data and annotate the cell types. This is already done from the previously published data set. And then they are using a feature selection algorithm. I'll briefly explain in a second. And then training machine learning classifiers. They use 4 machine learning algorithms: classifiers, random forest, support vector machine, MLP, and logistic regression. You are using these genes. And then you evaluate these classifiers on the test data set. So to look at the model training workflow, they have the pool in the panel A, they have the pool gene expression metric. The columns are basically the rows, the columns are basically the cells, and the genes represent, the rows represent the genes. And then these gene wells, basically for this the experiment that they have designed is they have alternate male and female genes, like the single nucleus that they have made in these gene emulsified wells. So the blue color, in the whole, all the figures you see, the blue color basically represents the males and the red one represents the females. So they have for the sex dependence gene expression to identify the differences. They have the way they have placed these one male, one female. So they have in this particular data set, they used three males and 3 female rats. So then after getting the pool gene expression metrics, they partition the data into training and testing set with 70 and 30%. So it's the similar way, alternate male, females. They have tried to balance the number of male and female cells in each the training and testing data set. They have tried to keep a balance. Then they have something called as the Borut-based feature selection method, along with like they do the differential gene expressions, but also like for the feature selection, they are using the Boruta method. And then after confirming the differentially expressed genes from with Boruta, they apply these models to the testing data set. And then towards the end, I will also show how they apply it to the another independent data set, which uses the NSC, single nucleus RNA-seq data set. So in this particular data set, the VTA data set, where they have over 22,000 cells, the panel B represents the UMAP for the 16 different cell types they have. And then the panel C is basically just showing the clusters colored with females and males. The red one is representing the females. Blue one is representing the males. And then in the D, they have tried to show the proportion of the different 16 cell types they have, proportion of the male and female cell types they have. So like they did try to keep a balance between how many females and males are there, the composition for the 16 cell types. Sorry. And then the panel E and panel F just show the clusters with a different number of cell types in the different number of clusters in the training and the testing data sets for the VTA data set. Then just like a brief that the Boruta is basically a feature selection algorithm, and it identifies significant importance for the cell selection classification for the VTA training partition. And then the Boruta adds the shadow features, which is shuffled copies of the original features. So like after like for the feature selection and the model performance, the training data set, which has 16 cell types and 23,000 genes, they identify 408 significant differentially expressed genes with adjusted p-values less than 5%. And then these differentially expressed genes were passed to the feature selection, which confirmed of the 408 differentially expressed genes, 285 differentially expressed genes. And then like after this, they in the panel B, they show genes ranked by average to fold change for different cell types. And then like OPC is basically here showing like higher gene ranks for females. And then in the panel C, they have this importance, like genes ranked by the median importance. So this is the importance values, the median importance values obtained from the Boruta's feature selection algorithm. So the feature selection algorithm shows confirmed, rejected, and shadow cell types. So the ones colored as red are basically the rejected ones. And on the right-hand side, these are the ones which Boruta algorithm has confirmed as differentially expressed genes. And here, like a tiny blue color is basically the shadow genes that Boruta defines. And here there are like 4 genes which are highlighted. These are the genes based on like the chromosome X and chromosome Y genes. And then these are like the highest ranked genes, and the FAM210 and TRABD2B, these are the lowest ranked genes. In panel D, this is the Violin plot representing different genes, expression level of different genes in the 16 cell types. So the highly expressed genes, which you can see in panel C, these 4, these are the highly expressed genes. So PY and ZIST, the ZIST is basically associated with the chromosome X, And this you will hear in the later presentation a lot. So these basically shows the expression level of different of these 6 genes and these and the corresponding to these 16 cell types. Following this, interpreting the different model features. So where we have the pool gene metrics, the columns representing the cells and rows representing the genes. So they have made four machine learning algorithms, namely the logistic regression, which is like linear and depends on the different classes. The 2 different classes where we have the Y, the males and females. Support vector machine, random forest, which has different subsets. It forms like different decision trees. And then the multi-layer perceptron, which is like which has like multiple layers, and then the nodes interconnected. And in addition, like to these 4 models, they also have two non-machine learning algorithms, namely the ZIST and the chromosome Y. So ZIST has basically the feature information of the related to the females, that is chromosome X, and chromosome Y is with the features related to the males. So this is how the ZIST is defined. Like the summation of expression levels for ZIST is like if the males, it is defined as male if 0, or female if it's the expression level is greater than 0, and it would be the opposite for chromosome Y. For the males, the expression values would be greater than 0, and females it would be 0. So these are the 6 different models that the authors have used. And then in the panel B, we see the sensitivity versus like the 1 minus specificity plot. Basically, this is in this whole paper, they have evaluated the methods based on the area under the curve or the area AUC, ROC curves. They have evaluated on the basis of the values obtained from this. So here sensitivity, like we have like the true positive rate or the false positive rate, and then the 45-degree line over here is basically where we have true positive rate equal to the false positive rate. And then here and the values we expect for a good model is to have higher values on the y-axis. So we see that compared to the 4 machine learning models, these are the ZIST, which is yellow color. So this particular model performs poor, and then also like the chromosome Y here, here we can see it comparatively performs poorer than the 4 machine learning algorithms. So here, if we look at the table below, the logistic regression and the SVM, they have overall AUC, like the highest AUC of 0.978, and like the accuracy of 0.197 and 0.92. So these 2 models outperformed compared to all the 6. And then these are also like there is a slightly these values are slightly less in the random forest and the multi-layer perceptron models. But if we compare to these 4 models, we see that ZIST and chromosome Y have really low values. So for interpreting these model features, we see that the machine learning algorithms, have outperformed the these 2 non-machine learning algorithms. And in panel here in the panel C, where they are looking at the proportion of like the classification, how much correctly the males and the females were identified. So the Y, for the chromosome Y, this identified that it did not misclassify the female genes or the female cells, but 22% it misclassified the male around 22% it misclassified the male's genes. And then in the ZIST, we can see like it did not misclassify the male genes, but the female side, the ZIST incorrectly classified 60% misclassified genes. And like if we compare these two non-learning machine algorithms classifiers versus the machine learning 4 machine learning models, the machine learning models have higher proportion of identifying the correct male and the female genes. Then, like in the same figure, continuing the panel D, they also did like on the VTA test data set, the logistic regression prediction probabilities, where we see like on the y-axis we have the counts, and then on the x-axis we have the probability. So the predictions, the sex predictions are basically, they have defined as if the probability is 0, it is classified as male, and if the probability is 1, classified as females. And here in these figures, you can see like some regions, they have highlighted some regions, like the blue lines over here around the 0.5 probability. So it is like, this is like narrow region where they are trying to do the thresholding by excluding the genes in these particular regions. So they have like they have defined as the narrow region, which is within the probability range of 0.4 to 0.6, and wide is basically in the region between these red lines. So that is between 0.5 and 0.75. So the reason they are doing this is when they exclude, say, the narrow region genes, they see that there is 94% accuracy, like almost approximately they do lose around 5% of the cells, while for the wide range where they are excluding between 0.25 and 0.75 probability, there they see the accuracy of 96%. But in this, they lose around 13% of the data. And here in this table, where they have like the number of cells included, so they have like different thresholds. None is basically, they're not applying any kind of threshold. They're not removing any number of cells. So this is the number of cells included, and then the threshold, the narrow threshold and the wide threshold. So the number of cells included, and after applying the thresholds are these values: 6310 for the narrow ones, and then 5790 for the wide threshold. So in this, they see that the number when they are excluding, like in these threshold regions, the number of female cells were included in these low confidence cell regions. So in this, like we see in the narrow threshold, there are around 3,182 female cells that were included, and then 175 female cells were excluding after applying the threshold. And then if they apply the wide threshold, around 470 female cells were excluded. And so when they did this, to just like apply this threshold and see how the accuracy increases by removing the low confidence cells, they found that mostly these low confidence cells, there are mostly female and non-neuronal cells. So like there is, like I will like shortly show where they compare, because of this finding, they did try to compare neuronal and non-neuronal cells after a few slides. In addition to this analysis, they also looked at something called as they looked at the RNA counts, increased RNA counts per cells. Like they see like increased RNA cells increases the probability of correct classification for all the models. So in these figures, they use like a logistic regression of VTA test partition, cell UMI account, and the model classification. So correct is basically correctly identified, basically with associated with the higher value 1, and incorrect is basically 0. So here we have these 6 plots. The upper two shows the non-machine learning models, and the below 4 are the machine learning classification models. So they look at the different UMI counts. On the 0, like these black circles are representing the UMI counts, and this blue, like they have these dashed red lines which indicate the RNA count corresponding to the 95% probability for the correct classification by any model. And then here at like this blue curve is basically showing, sorry, the blue curve is showing the logistic regression fit and shows the relationship between the UMI count and the corrected classification probability. So here at this intersection, this is like the minimum UMI counts required, like this intersection over here at 95% with the blue line and the red dotted line is the minimum number of cells required to attain the 95% classification accuracy. So for the compared to the non-neuronal, non-machine learning methods, which needs at least 6,000 in the chromosome Y method and 20,000 for the ZIST method, which is a very high number compared to all other methods. So these machine learning methods achieve 95% classification accuracy at a less number of UMI count, like around 5,000 for the logistic regression or the SVM method, and then MLP has around 6,000 genes to attain the 95% accuracy. Then here I am showing the neuronal versus the non-neuronal classification. So the gene metrics, they formed like they had made their VTA experiment, just that they have the neuronal and the non-neuronal. So here we see the nuclei arranged as alternate male and female cells, alternate male and females, blue, red combination. And then these cell types, then these then pass to the Boruta for the feature selection. And then again, they apply the machine learning models on these, like for this comparison. So they see that here, like as expected, the ZIST, which is a non-machine learning method, it comparatively performs poorer than the other methods. But compared to the neuronal and the non-neuronal, the chromosome Y method, we can see it performs poorer in the non-neuronal region. Here, like just I forgot to say that they are using 55 of the 70 neuronal and 232 of the 363 non-neuronal tags. These are identified after the feature selection from Boruta, the 55 neuronal and 232 non-neuronal differentially expressed genes. And then they look at the density versus the number of genes in the neuronal versus non-neuronal genes. And then in the panel D, they look at the mutual information, that is like the difference between the mutual information between the neuronal and the non-neuronal versus the genes ranked by these mutual information. So in this plot, the values which are lower than zeros, these provide information about the non-neuronal, and these higher ones, the information about the neuronal cells. And the ones at the very extremes, these are basically amongst the 6 genes which were identified as the extremes in the models, in the violin plots that I had previously shown. So these are the same 6 genes which are found on the extremes. So here they were trying to identify the reason if the performance of the non-neuronal is due to like the imbalance of the number of cells in these categories, but they see that like uh, the machine learning models, they show the accuracy, like the difference between the accuracy of the neuronal and the non-neuronal is like very low, around like 0.2, 0.3 in these values. So the classification in the non like the it's not limited by the in the non-neuronal cell is limited by the quality of the signal in the transcriptomes, but not by the imbalance of the way the experiments have been designed. So like apart from this, they tried another data set application, applying these models to a different data set. So this is also the information the data set they are using is from the previously published work about the NSE data set, which has around 39,000 cells. And they also have 16 cell types. In this, they are using around 8 males and 8 female rats. So in the panel, it basically shows the UMAP with the different cell type clusters, and B is the clusters colored by male and female. And then again, as we saw the results in the VTA data set, here also they find that the non-machine learning models perform poorer, while their 4 machine learning algorithms perform better compared to these 2 models. And then similarly, they have the proportion plot, like proportion, and comparing with the 6 methods they have applied. So we see that these 4 methods perform much better in predicting the correct male and female gene sets. So like even if in the table we can see that the overall AUC, ROC values and the accuracy for the 4 models are higher, and the RF methods AUC, ROC value is the highest amongst all the 6 methods. In conclusion, the results demonstrate that feature selection and the training strategies did not overfit the models to the VTA dataset. Models can be generalizable to cell sex classification of distinct dataset from different brain region. Also, here in this methodology, it does not require any additional experimental labeling or pre-processing. But currently, this is limited to the binary sex classification and might need adaptation for use in the different complex or the heterogeneous tissues. Thank you.